The overall goal of this experiment is to generate next-generation sequencing libraries from both the DNA and RNA of small populations of cells without splitting the sample at any point.
This method provides researchers with a new approach to study the structural variations of DNA on the expression of genes.
When we first thought up Gel-Seq, there were no protocols for simultaneously sequencing both the DNA and RNA from the same sample. Fortunately, there are now 3 published approaches that solve this problem.
The main advantage of this technique is that both genomic and transcriptomic information can be obtained from the same set of cells for negligible added cost.
Gel-Seq was originally developed with upright cassettes. However, this protocol can be adapted to work with any standard gel electrophoresis cassette. Prepare the filler, the high-density, and the low-density precursor mixtures in 3 tubes. Add the reagents in the order described and vortex the mixture. Do not prematurely add either APS or TEMID. Wait until directed. Next, degas each gel monomer solution by first inserting a needle through the cap, then connecting the needle to a house vacuum line and submerging this assembly in an ultrasonic bath. Wait about 1 minute until no more bubbles emerge from the liquid. Next, to the filler precursor mixture, add the TEMID and APS and vortex briefly. Within 3 minutes, use a 1 milliliter pipette to add 6 milliliters to each gel cassette without spilling. Ensure the fluid is level by positioning the cassettes upright on a level table. Then, gradually top off the cassettes with degassed ionized water, being careful to minimize the mixing of the polymerizing mixture with the water.